Hello, my name is Dr. Alan Ramsay. My email is here, and it's a great pleasure to speak today. Thank you very much for the invitation from the Argentinian Society of Hematology. I'm very sorry I cannot visit Argentina, but I'm delighted to give this presentation. So I'm going to talk about the mechanisms of resistance around immunotherapy for CLL. So this slide just gives the following disclosures. The Institute receives research funding from the two companies listed here. So studying the tumor microenvironment is a major interest in the group, and the idea is to optimize or develop effective immunotherapy for CLL, but also other lymphoma types. So certainly developing combination immunotherapy represents a very compelling option to overcome resistance to current therapies, as well as deepen the responses to even some of the newer agents that we're working with. Now, when we think about immunotherapy, clinical trials have shown that anti-PD-1 antibody alone, as well as the CAR-T trials to date, are showing lower response rates than we would like. So this clinical experience tells us that the tumor microenvironment in CLL, and this applies to other lymphoma types, may be or is presenting a barrier to this type of therapy. So my group is very interested in studying the obstacles that cytotoxic T cells, as well as other immune cells, face in what we call the tumor microenvironment. And the idea is to then overcome these barriers to optimize therapy. So today I'm just going to give a very brief snapshot summary of some projects which are currently running. So these are around studying patient T cells, as well as fibroblasts, which are more understudied in the microenvironment. So the first one is immune monitoring studies from clinical trial samples. These were frozen samples from patient peripheral bloods, and we've been studying the T cell compartment, and this is during ibrutinib-based therapy. I'll also describe a project where we demonstrate that targeting T cells with an immunostimulatory drug can potentially turn the microenvironment from cold to hot and then sensitive to immunotherapy. I'll also describe preclinical work studying T cell engaging bispecific antibodies. And lastly, I'll describe the study of lymphoid tissue fibroblasts, which we're showing become activated in CLL, as well as other lymphomas. And they have altered immunoregulatory function, and this has implications for immunotherapy. So just to introduce, really, a lot of the audience will be aware that there's complex crosstalk between the malignant B cells in CLL and other surrounding cells, including the immune T cells. So this is actually meant that in the field, T cells have been regarded as what we call a double-edged sword. So for example, a lot of literature provides evidence that activated helper T cells, the CD4 compartment, can actually have a tumor promoting capacity in CLL, which is not surprising given the B cell nature of the malignancy. On the other hand, chronic exhaustion, T cell exhaustion, is known to contribute to tumor evasion from the cytolytic activity of the killer T cells, the CD8 T cell compartment. And indeed, my own work previously has shown that T cells from CLL patients have an impaired ability to form what we call the immunological synapse, which is the signaling structure between T cells and antigen-presenting cells, or indeed tumor cells, when the T cells are interacting. So a major question is, how can we engage anti-CLL cytolytic activity? So I'm very interested in activating the killing function of T cells in patients. Now, let me start with the first project. So this has been this is just illustrates the type of work we're doing. We've been studying the E1912 trial in collaboration with the Mayo Clinic, with Dr. Neil Kay, Dr. Tate Shanafelt. Now, this trial compared rituximab with ibrutinib, a combination compared to traditional chemoimmunotherapy. And we've been studying longitudinally the T cell compartment from patients which were enrolled in the trial. And these patient samples were biobanked, sent to the lab here for our T cell functional analysis. But here's the flow cytometry data. And you can see that in agreement with other studies, we see that ibrutinib-based therapy is actually reducing T cell numbers in patients. And this includes the naive as well as the effector subsets of T cells. Now, because this is a major clinical trial and the clinical data is coming together, we are also interested in investigating the relationship between immune cells and clinical responses. So here, what I'm describing is that we found a significant association between the number of T cell subsets and, in this case, progression-free survival. So at baseline, this was particularly evident. We found that patients which had increased numbers of effector CD8 as well as CD4 T cells, this included the exhausted T cell population, so PD-1 positive T cells. Actually, the increased numbers of these T cells associated with improved progression-free survival during ibrutinib rituximab. So this evidence really tells us that an active but yet exhausted T cell response associates with better response to this type of therapy. At 6 months, we found the increased numbers of CD8 T cells as well as NK cells was prognostically favorable in terms of PFS. And interestingly, at the later time point that we investigated, 12 months, we didn't find any association between T cell numbers and clinical responses. So we've also been looking at the T cells functionally, because the ability of a targeted drug to improve effector killing could actually allow a gateway for immunotherapy. So here we've been performing the cytotoxicity assays in the lab here. And again, longitudinal functional analysis has shown that T cells at 6 months or 12 months during ibrutinib rituximab are actually showing enhanced killing against the baseline CLL autologous clone. And that's shown in this graph here. And interestingly, at the later time point, 12 months into treatment, a reduced cytotoxic capability was actually significantly associated with increased infections, so grade 3 infections during ibrutinib rituximab therapy. So we also, because of this trial was also comparing to traditional chemoimmunotherapy, we also assayed T cell function on the FCR arm. And this actually revealed that the T cells from these different time points, 6 months and 12 months, actually failed to elicit any significant tumor killing. So next, we wanted to explore the addition of immunotherapy. So here, ex vivo, we're treating with anti-PD-1. And interestingly, here with the ibrutinib rituximab arm, we saw no improvement in T cell killing when we treated the ibrutinib rituximab T cells with anti-PD-1. In contrast, we did see activity with anti-PD-L1, which is the inhibitory ligand, which can be expressed on T cells as well as CLL cells. So just thinking about the mechanism, we've been looking at the immune synapse. And you can see that during ibrutinib rituximab on the left, we found that the therapy is actually enhancing the ability of T cells to form synapses. This contrasts with the traditional chemoimmunotherapy, where synapse was not being improved during therapy, post-therapy. And then when we actually look at the types of T cells which are engaging the CLL cells, what we noticed was at baseline, the helper CD4 cells were forming more synapses with the CLL clone compared to CD8s. And when we then compared this to the clinical data, we found that it increased CD4 synapses actually correlated with poor progression-free survival during ibrutinib rituximab. Interestingly, during IR, ibrutinib rituximab, we see at 6 months, 12 months that there's a switch in synapses, and the CD8 T cells are engaging the tumor more compared to CD4. And we believe that this is in keeping with the improved cytolytic activity, which is mediated or promoted during this targeted therapy. Okay, so at this point, I've just included this slide here because it just shows, well, what is the challenges or the resistance mechanisms which the T cells or immune cells face within the CLL microenvironment. So what we believe is that the microenvironment is non-inflamed. And what does this mean? It means that there's a very low number of cytolytic T cells, so very low number of CD8 cells. You can see that in this multiplex tissue staining that we've done here. That in CLL, there's a very low number of diffusely infiltrated CD8s. And this contrasts with a much higher number of activated CD4. And what we always do in our assays is we compare CLL to, for example, reactive or healthy lymph node tissues. So there's a low number of cytolytic T cells, and we believe a biomarker is PD-L1 expression, so a very low tumor expression. The CLL cells are not expressing PD-L1 at high levels. If anything, the T cell compartment is expressing PD-L1 as shown here. But again, it's very low expression compared to other cancer types, the solid cancers, which are sensitive to immunotherapy. So this brings me into the second project, which really is proof of principle that immunostimulatory drug, in this case, a CELMoD, which is an IMiD type drug, next generation drug. And we've shown that avadimide can stimulate immune synapses very strongly when we are assaying CLL patient cells. So the T cells are forming the synapse. The checkpoints, PD-L1, are actually upregulated in terms of expression, and they're localizing to the synapse contact with the tumor cell. And we've tested this in treatment naive, and this is the cytotoxic killing assays. You can see, hopefully, in this busy diagram that avadimide on its own can significantly improve killing function, allowing the T cells to kill autologous tumor. The anti-PD-1 alone is ineffective, but when we pair anti-PD-1 or anti-PD-L1 with avadimide, we're actually seeing maximally enhanced killing function from the T cell compartment. And then going back to the trial with the Mayo Clinic, we find that these immunostimulatory drugs can actually boost the T cell killing response when we treat T cells which had been previously exposed to ibrutinib rituximab. And that was both at the 6 month and 12 month time points. So just mechanistically here, we've performed RNA-seq, and specifically to do this, we purified T cells from patients and then treated these T cells with different types of immunotherapy. And what we find is that avadimide is actually promoting the interferon alpha and gamma type 1, type 2 gene response signatures. A very strong inflammatory response is triggered in the T cells. And when we break that down into functional pathways, you can see that chemokines, cytokines, the F-actin regulatory pathways, T cell proliferation are all significantly upregulated with avadimide. And this is kept when we have the combination with the checkpoint blockers. And hopefully you can compare that to anti-PD-1 alone or anti-PD-L1, which are having much less impact on the exhausted T cells. So this really illustrates, hopefully, to you that we need very potent immunotherapy to overcome T cell exhaustion. And I don't have time today to describe this, but really what we're seeing is that this avadamide is actually normalizing the interferon pathways in patient T cells, which are deregulated in the treatment-naive setting. So the group here also run xenograft assays. So again, the advantage here is we're using frozen PBMCs, and we can successfully engraft in NSG mice. You can see here that in untreated mice, the human CLL B cells are growing in the spleen. They're proliferating. The CD4 helper T cells are engaging the tumor and are critical to allow disease establishment. The CD8 cells are in green. Hopefully you can see that, and they're actually showing an excluded pattern. So this is a powerful tool, we believe, then to model, ask various questions, including novel treatments. So avadamide is the drug that we've been testing here, and you can see, hopefully, that when we treat the mice with avadamide, that the CD8 population has proliferated, it's expanded, and it's starting to actually cause the tumor to regress. Anti-PD-L1 or PD-1 was ineffective as a monotherapy. So that's in keeping with the human trial data. And the combination is most effective with very low residual tumor cells left at the time point that we chose to study. So, and I've just included this image here. This actually shows that the avadamide is triggering chemokine expression in the T cell compartment. And we believe that this actually this sends very important cues to enhance the trafficking of T cells into the tumor area, which has relevance for overcoming the lack of CD8 movement as a barrier, as a resistance barrier in CLL. So this just summarizes this particular project. We show that avadamide can trigger the type 1, type 2 response. Importantly, this increases T cell numbers. It releases inflammatory genes, proteins, which are needed in the microenvironment to get T cells to move, to form synapses. And this can pair very effectively preclinically with the checkpoint inhibitors. Okay, so now I'll just introduce the third project that I'd like to summarize today. This is a newer type of immunotherapy that we've been working with. This is T cell engaging bispecific antibodies. So we've been working with glofitamab, which is an anti-CD20, CD3, it's a CD20, CD3 T cell bispecific. And what it does is that it allows T cells to bridge and to form very strong synapses with CD20 expressing CLL cells. So this glofitamab is being tested in clinical trials in non-Hodgkin's lymphoma, but here we're doing preclinical work in CLL. Now, when the bispecific has engaged T cells and the tumor cells, it sends a very potent activatory signal through the CD3 receptor. And what we've discovered is that it's a very strong stimuli for activating these previously exhausted T cells. So here we see that the proliferation function of the T cells is significantly improved in both the CD8 compartment as well as CD4 cells in CLL. We see, importantly, when we look at cytotoxicity assays, that the TCB can promote the ability of T cells to kill autologous tumor very effectively. And interestingly, we also find that the TCB can convert helper CD4 T cells to become killer CD4 T cells. So there is cytolytic function coming from the CD4 cells, but this is mainly CD8 in the assays that we're presenting here. We've also been looking at patient samples which are responding or refractory to ibrutinib. This is in collaboration with Toulouse, Dr. Loïc Jesbart and his colleagues. And we're seeing here that the TCB, CD20 TCB, is very effective at getting the patient T cells to kill residual disease when patients are on ibrutinib, either responding or refractory to the targeted drug. And this is, I don't have time today, but we can see that the TCB is superior to many other therapies in the lab, at least, and including anti-PD-1, which I'm not showing here. Okay, so we're also interested in exploring what are the resistance mechanisms to immunotherapy. And this is where our interest in the microenvironment, this is the reason why we study the microenvironment. So here we're just demonstrating that nurse-like cells, so these are well-described microenvironment components. When we grow these in the lab, we can then perform our immunotherapy assays using T cells and CLL cells from the CLL patients. And what we find is that the presence of nurse-like cells is actually suppressing the proliferation of the T cells in response to the TCB. So we believe that this is a resistance mechanism potentially in how a TCB would work if you were giving this to a CLL patient. So these nurse-like macrophages are suppressing proliferation. We've also been studying the endothelial cell compartment, and I don't have time to describe the tissue staining, but effectively when we model this in the laboratory, again, we're assaying the ability of the TCB to work in the presence of this tumor microenvironment cellular compartment. And here, this is cytotoxic killing, and we've screened many primary samples. And what we see is we can dampen the ability of the TCB to work, significantly work, when we are using the drug in the presence of endothelial cells. So this endothelial cell compartment is suppressing T cell killing function following immunotherapy. Now this then has generated interest in combination immunotherapy. So here we're testing the ability of what we call a tumor-targeted agonist drug, in this case, CD19-targeted 4-1BB agonist. And we're testing the ability of this drug to be paired with a T-cell bispecific. And the idea is that combination can counteract the suppression or the dampening of how microenvironment cells are reducing the TCB reaction. So the reason why we're looking at 4-1BB as a target is that when we treat the patient samples with the T-cell bispecific alone, you can see that T cells start to express this co-stimulatory receptor, 4-1BB. So then when we pair the TCB with the CD19 4-1BB ligand drug, you can see that we see a significantly additive tumor killing in our cytotoxicity assays. So then we go back to the microenvironment models, and here we're showing that when we use the combination therapy, we can increase the proliferation of CD8 cells when we're using combination compared to the TCB alone. Going back to the endothelial microenvironment assays, we can see again that the combination treatment can overcome this dampening the dampening ability of ECs to suppress the TCB reaction. So really, this is just preclinically studying the microenvironment as a resistance mechanism and showing that perhaps with the right combinations, we can overcome and optimize immunotherapy in the microenvironment. So we've also employed the xenograph model here, just very briefly because of time. You can see that the TCB reaction drug, in these mice has allowed CD8 cells to start expressing 4-1BB following treatment. When we give the TCB, it's partially effective at causing tumor regression, but with the combination therapy, we see maximal tumor killing. In this case, the tumor has been labeled using a red fluorescent dye, and you can see that with combination, there's very minimal red remaining. So we also look at the ability of these drugs to activate the T cell compartment, and you can see here that it's a lot of CD8 cells have proliferated, and they're starting to express much more granzyme B, the cytolytic molecule, than compared to the treatment-naive cohort. And this is a very small diagram here, just to show you that in keeping with avadamide, the T cell bispecific can also increase expression of these chemokines, for example, CXCL10, that we believe is important for inflammatory T cell recruitment signals when the mice are under treatment. So we can quantify this either by flow or imaging, and you can see that combination therapy is significantly reducing the CLL disease volume in the spleens of these mice. Okay, so this is just another assay here. This is actually using fresh lymph node biopsy. So these are diagnostic lymph nodes from suspect lymphomas, which we then have an ethical and consented approval to study for research. Here we use a vibrotome to make very thick cuts of fresh viable tissue, and then we can then study these in the laboratory. So here we've been treating the organotypic slices with the drugs that I've been describing, and you can see that this essentially patient microenvironment tissue, the TCB is eliciting a partial response. But when you would use combination therapy, we see maximal tumor cell killing. And we're using caspase 3 as a marker for apoptotic CLL cells. So this is a CLL lymph node, which was just diagnosed at the same time as this type of work. And you can see that the immunotherapy is effective in these organotypic assays. We've also been lucky enough to assay relapsed CLL patient tissues. Here, we're looking at ibrutinib relapsed, but we've also assayed venetoclax relapsed lymph node tissue samples. And you can just for proof of principle, we can see that these combination T cell bispecifics are working in high-grade lymphoma, as well as the more indolent follicular, and of course in CLL, which we've been describing. The advantage of the organotypic system is we can also look at the intact microenvironment. So here, this is endothelial cells within the lymph node tissue, and we can see that they're expressing the inhibitory ligand PD-L1. And this actually increases after TCB. So we believe that immunotherapy will trigger inflammatory signaling in all cellular compartments, including the stroma. This means that we could potentially combine immunotherapy with anti-PD-L1, anti-PD-1, but also the CD19 4-1BB agonist, which we've been modeling here as a co-stimulatory approach. So the last part of my talk is looking at the fibroblasts within the microenvironment. So you can see here the classical CLL effaced lymph node, and the T cells, which I showed earlier from confocal, are diffusely infiltrated, lost in this sheet of malignant B cells. We've been very interested at King's since I started my group here at really studying the fibroblasts. So these are the fibroblastic reticular cells, or FRCs, a very important immunological stromal subset and understudied in lymphoma and CLL. But what you should know is that fibroblast reticular cells have a very important role in regulating all types of immune cells in humans, for regulating a response to a viral infection, COVID, or indeed malignancy. So we're studying this in CLL. So what we do is we are growing primary fibroblasts either in 2D, 3D cultures, co-culturing them with tumor cells. I haven't shown that here, but we either do that in transwell or direct contact. So CLL cells with the fibroblasts. And what happens is that CLL exposed fibroblasts become activated, and they actually take on this stretched morphology. So that shows they become decontractile and elongated, which is quite a marked phenotype compared to the more contracted fibroblasts in the healthy scenario. You can see that primary fibroblasts from a CLL lymph node also take on this stretched activated phenotype. So we perform flow cytometry. We look at how fibroblasts can support primary cell survival, primary CLL cell survival. And we also measure, following co-culture, what happens to fibroblasts after tumor contact. And here we actually have documented a very important finding that we believe in relation to an immunoregulatory molecule. So in this case, it's podoplanin. The CLL activated fibroblasts have down-regulated podoplanin. We confirm this in patient tissues, a loss of this important molecule compared to healthy. We also are interested in the transgenic model, TCL1, potentially other models in lymphoma. And in the case of this model, we have evidence that the fibroblasts have also become activated in keeping with the human tissues above. So one approach is transcriptomics. Here we've done bulk RNA-seq on fibroblasts which have been exposed to human patient CLL cells. And I can't go into all the details at the moment, but we see that the fibroblasts become activated, and they actually start showing altered pathways which are linked to angiogenesis, extracellular matrix remodeling, cell adhesion, interaction capacity. So we believe that this concept of tumor activating fibroblasts within the lymph nodes has important relevance for resistance to immunotherapy, resistance to how T cells are actually mounting a response against the CLL proliferating clones. Right, I'm running out of time here. So this is just modeling in the lab, showing that we can show how dendritic cells are interacting with fibroblasts. You can see that dendritic cells in this movie here will spread and move very carefully across the fibroblasts. This is a slowed down movie. But in CLL, what we find is that the dendritic cells are not making effective contact with the fibroblasts. They're not spreading, and they have very stochastic movement, very random movements. And we can measure this in the lab. And then when we go back to tissue, we can see that in health, dendritic cells have very close association with fibroblasts that are moving around the lymph node. But in CLL, the dendritic cells are detached, are not interacting with the fibroblast cells. Therefore, we believe the dendritic cells are mislocalized. They're becoming what we say derailed. They're falling off the fibroblast track. And this has important implications for mounting an immune response, and we believe actually contributes to the immune deficiency that is characteristic of CLL, but also other malignancies of the lymph node. So looking at the T cell compartment, they also use fibroblasts to move around, to become compartmentalized and regulated. In CLL, the T cells become very sluggish. They become very slow. You can see it's quite different to the healthy scenario. We can quantify that from the microscope. This is CAR T cells moving rapidly on healthy fibroblasts, but here we have a very dramatic slowing down of how CAR T cells are moving on these activated fibroblasts. So we believe this is a resistance mechanism potentially to CAR T therapy, both for CLL but also other lymphomas. And we're very interested in pursuing this type of study. We always go back to patient tissues, either on treatment, post-treatment, or at treatment naive. And here in the human setting, we can see that the fibroblasts have lost expression of homeostatic chemokines, CXCL12, CCL21. So this will really have important implications on how T cells, dendritic cells are moving within the microenvironment. Okay, to summarize, what are the resistance mechanisms? Well, there's certainly profound T cell exhaustion and an impaired inflammatory response, low numbers of CD8, low expression of inflammatory markers that are known to be required for immunotherapy. So the challenge for us is to develop therapies that can work long-term for patients. And one of my major interests currently is thinking about how stromal cells, for example, fibroblasts, are contributing to resistance to immunotherapy, but also resistance to how immune cells are generating an anti-CLL response. And that's what I've just described with how the CLL-activated fibroblasts may be suppressing dendritic cell motility, T cell motility. And we've also shown functionally in the high-grade lymphomas that they can actually tolerize the killing function of CD8s. So lastly, a huge thank you to EHA, which I actually was successful with a fellowship at the start of my scientific career. I'd also like to thank all the collaborators listed here, which are international throughout the world, UK, Europe, US. And a huge thank you to you for listening. And I'm very keen to establish collaboration with your groups. So please contact me with my email and we can follow up, and I'd be delighted to have further conversation. So thank you very much.